Okay, now let's talk about Chapter 18, um, how the genome is regulated by organisms. We'll start with bacteria. Bacteria have, well, first of all, when we regulate genes, we often use what's called negative feedback, and that is where you essentially stop the process, there we go, um, from running, essentially using the final gene product. So in this case, we have a biochemical pathway that is responsible for making tryptophan and this amino acid. And there's a couple different ways you can have negative feedback. You can have the final gene product, tryptophan, inhibit an enzyme earlier in the process, or tryptophan can somehow regulate the expression of the genes. Either way is going to have a negative feedback and control the expression of those genes. Now, bacteria have their genes clustered in these things called operons. An operon is a cluster of genes that are related in the sense that they code for gene products that are involved in the same biochemical process. Um, the trip operon is one responsible for making tryptophan, this amino acid. Um, it is known as a repressible operon because the presence of tryptophan, what you're trying to make, represses the operon or blocks it. If we look here, here's our series of genes in our operon. There's also what's called the operator, um, and a section of of DNA that the polymerase will bind to, and this is collectively known as the promoter, and it's again the area where the polymerase goes. Further upstream of the gene is this gene of the operon is this gene that's responsible for coding for a protein known as the repressor. Um, in a repressible operon, the repressor is produced in the off state. It will not block the operon from being transcribed. But with the presence of the tryptophan, what it does is it binds with the repressor, activates it, and it binds to the operator and blocks the polymerase from doing its job. Thus, it represses the operon. It will not run. Um, Here is what's known as the lac operon, and the lac operon is a series of genes that are responsible for producing proteins that help with the digestion of lactose sugar, milk sugar. And it's known as an inducible operon because the presence of lactose, this compound you're trying to digest, induces the operon, causes it to run. Here, the regulatory gene codes for a protein that is going to control the operon, but it's produced in the active state, so it will block the operon. The presence of lactose inactivates the repressor such that it doesn't bind, and so therefore the polymerase is able to do its job and the operon runs, the genes are copied, you make the enzymes you need to digest lactose because lactose is present. So lactose induces this operon. Now, you can have further levels of control, and we see this with the lac operon. Um, Bacteria can feed on different types of foods, different types of sugars. So glucose, lactose are examples of some of these sugars. Um, and just as you may have preferences in what you like to eat, bacteria are the same way. They prefer glucose, and lactose will be consumed after glucose has been consumed. So what happens is if lactose is present, the lac operon will normally be induced and you'll copy those genes so you can digest lactose. But what about if glucose is present and the bacteria wants to digest it and consume it first? Well, what happens is in the presence of um, glucose, you've got lactose present, so the repressor is not operating and the operon should run. In the presence of glucose, you have this transcription factor that is in an inactive state. And without the transcription factor to assist the polymerase, even if the operon is not being blocked, you will not transcribe those genes. Only if you have lactose present, the repressor is inactive, and you don't have glucose or you have very little, this transcription factor becomes active and helps polymerase to transcribe the genes. So this is sort of a second level of control that you can have as to whether genes will be transcribed. All right, now with eukaryotes, it's a little more complicated. We have the DNA housed inside the nucleus. We have RNA transcripts that are processed to remove introns. Um, and so there are other stages at which we can regulate this process. Um, Again, you can just block transcription or you can let it go. You can control RNA processing, um, transport of the RNAs outside of the nucleus. You can control the translation process, the making of proteins. If proteins are no longer needed, you can degrade them. When mRNAs are no longer needed, you can degrade them. So there's many steps. So first, to make the DNA more accessible for transcription, you can do what's called histone acetylation. You remember histones are the proteins that DNA are wrapped around, and they form these things called nucleosomes. And the histones have these little tails that stick off the side. And if you add acetyl groups to them, that keeps the DNA from sort of clumping together, from condensing. So the DNA is spread out and it's available for transcription. On the other hand, you can do what's called DNA methylation. You add methyl groups to the cytosines of DNA, and that binds the DNA up and doesn't allow it to be transcribed. So you can basically make it available or turn off the DNA. And this process, particularly DNA methylation, is something that's relevant to this phenomenon what's called epigenetic inheritance. And it's where the genome gets modified in a way that can be inherited that is not due to, say, a mutation of a gene, a mutation of the DNA. That's the typical way we think of traits and modifications of traits being passed on as a change to the base sequence of the DNA, and that's what we would talk about with inheritance in terms of Mendelian inheritance and evolution occurring is these mutations. With epigenetic inheritance, what happens is the DNA can be bound up, say with DNA methylation, so that a particular one of your particular alleles is not used or not expressed. And normally we would think of those as being opened up again from one generation to the next. But with epigenetic inheritance, this trait that's been modified in a way other than a mutation can be passed on from one generation to the next. Sometimes this happens in utero, where there's a modification of the germline, essentially the reproductive cells and their DNA, and that can be passed on from one generation to the next. Other times it's, and this is an image I got off from a Nature paper. Here's the reference to it if you want to take a look at it. What they call experience-dependent inheritance, and this is where after an organism is born, these baby mice essentially, the degree to which the mother, in this case, cares for them can modify their DNA, which can be passed on. And again, these are modifications that don't have anything to do with the base sequence of the DNA. All right, so in eukaryotes, regulation of transcription, here we're looking at, so we've got a gene with different exons and introns, a promoter where the RNA polymerase will go, Upstream, we've got these control elements, these sections of DNA that, as we'll see, are going to be involved with these compounds that promote and facilitate transcription. Um, we know that in eukaryotes, the primary transcript, the initial RNA that's made, in this case this mRNA, with these exon and introns is going to be processed, and the introns are going to be removed. Cap and tail are going to be added to produce our final transcription. Um, so these processes all have to happen in order for um, genes to be made use of and expressed. In order for the mRNA to be made, for the transcription to occur, for any RNA for that matter, and transcription, you have to have certain compounds around. You have these transcription factors, these proteins, um, and we talked about the enhancers above. You have these compounds called activators that bind to these parts of the DNA that are upstream of the gene. Um, and what happens is in the presence of the activators and the transcription factors, they clump together, and apparently this is what happens. The DNA literally bends, and you get what's called the transcription initiation complex. And this whole thing, combined with RNA polymerase, basically initiates the process of transcription. So if the transcription factors or the activators are lacking, then that's another way of not expressing the gene, not transcribing the gene. Enhancers are what give you some specificity of which genes are expressed in which cells. So again, all of your cells contain the complete genome and all the genes necessary for you, but only certain genes are used in certain cells. Um, so, for example, albumin is a protein that's made and it's needed in liver cells. And so you have the activators that will allow that gene to be expressed. Um, but inside your eye, you have different kinds of activators. Um, and these are not ones that cause the expression of the albumin gene, for example, but a different kind of protein called crystalline, which is something you need in your eye. And in your liver cells, you don't need that. So the activators can help specify which genes are being expressed in which cells, and therefore which proteins are being made. Um, so you've got your DNA inside the nucleus. It's kind of spread out in the chromatin state, and you're going to transcribe different parts of it. Um, and so bacteria have their relevant genes for a particular process clustered into these things called operons. In eukaryotes, you don't have that. A gene that's needed for a process, or genes, I should say, might be on different chromosomes. You need these 4 genes to be transcribed, but they're on different chromosomes. What happens is there can be a physical coming together of those portions of the 4 different chromosomes into what is called a transcription factory, this region inside the nucleus where you'll be transcribing those essentially all at the same time. Again, not exactly an operon, but equivalent to an operon, where you're going to produce the relevant proteins that are needed. Um, okay, so after transcription, you've made your RNA, and so we've got these different exons, and there's this process we call alternative RNA splicing. So we've got these 5 exons, and you might use all 5 of them to produce a particular RNA and then a particular protein, or you might splice them together in different combinations. So from one gene, essentially, you can put the exons into different combinations to result in different kinds of proteins. Almost out of time here. Hope to get through this section, but of course, then you can break down mRNAs when they're no longer needed. Certain proteins can either favor or block the process of translation. And then finally, proteins that are no longer needed can be marked with this compound and then degraded in this structure called a proteasome.